aim to show you today that biophotonics is widely present in our daily lives already, and it has many applications in both micro and macro scales. It is widely used for disease diagnosis and treatment, and it has potential to revolutionize healthcare. So if you have gone to the hospital, you may have seen an instrument like this. The doctor clips your finger with something, and then you just have your blood oxygenation and heart rate after that. This equipment has a red light on it, and it will measure the absorption of oxygenated and deoxygenated blood and how this varies with time. So this instrument has been already portabilized. It can be accessed with your smartphone, or you can see this technology already present in smartwatches, for example. But this is not the only technology that measures something related to blood. So blood can be, for example, in a crime scene, and it can be seen with microscopy, for example, in forensic microscopy. You can see, you can investigate further crime scene with blood analysis, but also with other objects present there. Microscopy can also be used for other applications, such as biofluid diagnostics. There are not only applications with blood tests, but macrourinalysis. It is possible to have analysis if you go to the laboratory and they collect your urine, and then they will actually check for the sediments of that urine and see several diseases, or probe for infections that can be of many kinds. So like either viral infections, yeast infections, or parasitic infections. Now, if you're looking for decontaminating and disinfecting environments because to avoid infection, then you might see some technologies for ultraviolet that you can use to decontaminate hospital rooms or any surface that you may have contaminated in a certain process. So this is very widely used in the hospitals, and it actually has been more and more common because of the COVID outbreak, as you may have heard of. So, so with this technology of surface disinfection, we can also decontaminate the surface of fruits and nuts, for example. And biophotonics is not only used for this kind of applications, but also for imaging inside the body. So there are many technologies where you can image from inside all your gastrointestinal tract. You can look for ulcerations, irritations, or even cancer. So you can use endoscopy and colonoscopy for this this application, and you may have relatives that may have gone through these procedures. So biophotonics is also used to image during surgery. So it is used for surgical guidance, especially for having the surgeons to not remove vital structures such as important blood vessels or nerves. So it's very useful for guiding the surgeon during the procedure. And if we are talking about all this surgery, biophotonics can not only image inside the body, but with the advent of laser, there were many procedures that were available that were before they were unthinkable before the advent of laser. So some of these procedures are eye surgery, for example. So the most common one is LASIK, so the laser-assisted in situ keratomiliosis. And this is a procedure that is very common. If you just get the statistics in US, there are 700,000 procedures performed each year. And this is actually a decrease from the last years. So it's a very common procedure already. And obviously in US, it has 10% of the population has serious vision limitations or blindness. So there is a huge market there, but there's technology that is even expanding even more to other countries in the world. So not only for eye surgery, but there are other procedures that uses laser. And for example, lithotripsy. In this case, laser is used to break kidney stones that are blocking the urine passage from your kidney to your bladder. So usually the patients can't urinate, and the because of the blockage caused by these stones, the patient actually feels a lot of nausea and pain. So it's a lot of discomfort there, and they actually need to go through a surgical procedure to break these stones. And there's a very common problem, like the lifetime incidence is around 10% for and all the people. And once you have it, then it has 50% of recurring after 5 years and 80% of after 10 years. So biophotonics actually helped to solve a problem that was very recurrent in the world. So not only that, biophotonics, not only for procedures where we use laser with a high intensity and a very localized spot, but sometimes we use light that is spread out over a large area. In this case, we have a light therapy with photobiomodulation. And photobiomodulation is used for many applications, such as aesthetics. So you can have acne or wound healing. It's also used for sports medicine, so increasing the performance of athletes and healing their injuries after sports. It is also possible to have pain relief and some treatments for anti-inflammatory response, open vasodilation. So photobiomodulation is used for a number of applications. This is the list map of where it is applied and which diseases and disorders that are treated with photobiomodulation. In this case, these are the treatments where you want to have an effect that you modulate the biological response in the area you illuminate. But if you want to be even more specific, so if you want to target specific cells, what you should do is you need to sensitize the cells first, so make them more sensitive to light so that you can kill specific cells, for example, bacteria or fungus or even cancer cells. So this procedure is called Photodynamic Therapy, or PDT. It's also a very common procedure in biophotonics. So this is a map just for the human infections, the treatment for human infections that are already clinically approved. So these are the lists of what is already daily used in the clinic for infections. There are other applications for cancer and a lot of applications that are being researched. Just to give you an idea of what are the results that are achieved for one type of treatment. If you see a nail that is infected with fungus in your left side, and then you see a healed nail in the right side, you see that the results are amazing. So photodynamic therapy is actually widely used, and it's very known. It's one of the huge successes of biophotonics, and this is the reason why it has been so researched and also used worldwide. So it's not only this the only source of the success of biophotonics. One of the most successful methods for imaging is the retinal, what you see in the retinal exams. So the retina is this light sensitive layer that you have in the back of your eye. And you may, once you go to your ophthalmologist, you may see him or her asking you to look to an equipment like this. You should see an image very far away, and then you focus on that and don't move your face. So what they are doing there is actually looking at the profile of your retina. And by looking into these, they can see, they can probe for certain diseases that are occurring, not only the ocular diseases, but other diseases that may have an effect on their retina. So this technique is called optical coherence tomography, and it is widely used. It was one of the another huge success of biophotonics, especially in medical imaging. So in terms of medical imaging, biophotonics in medical imaging, and just in medical imaging, biophotonics has already a contribution of a share of $73 billion in the medical imaging market. So we usually we are used to hear when our doctors, when we go to the doctor, they will prescribe you like you should go to do X-rays or make some ultrasound or magnetic resonance imaging, or maybe if you have cancer you may go through radiotherapy. But the global market for the radiological world is actually much smaller than the light-based techniques or the optical techniques. So actually the biophotonics contribution to medical imaging technologies is huge, and it's 64%, or as I said, $73 billion of the medical imaging market. So to see why the biophotonics is so successful, let's understand just a bit of the basics. So just quickly going about the electromagnetic spectrum. So you may remember that the wavelength is the minimum distance that you have for repetitions of a wave shape. And just if you see the visible spectrum, then you may remember that at 400 you have violet, violet, and at 700 nanometers you have red. If you go to wavelengths shorter than 400, then you have ultraviolet, X-rays, and gamma rays. After 700 or red, you have the infrared and radio waves. So if we are looking at the electromagnetic spectrum and we consider the medical imaging technologies, there are actually three ranges where the medical imaging technology most falls. So this is the X-ray imaging, the X-ray range, the optical range, and the magnetic resonance range. So these are actually the windows where we have highest transmission for water, and the highest transmission is actually found in the optical range. Uh, Another thing that is special about this optical range is that um, the most of the useful molecular information is actually optical. So if you see the changes that are occurring because of a disease, um, once these changes um, are related to a higher concentration of biomolecules that are changing, it's very easy to detect, as you can see in the figure here to the left. So once you see, for example, structural changes, this means that a lot of molecules are being changed, and you see the structural changes. Once you decrease the concentration of these molecules, it's more difficult to detect the disease, and it is more difficult to detect it early. So you once you decrease, so you can have high concentrations of changes. So with you see structural changes. Once you have changes at a smaller concentration, you will see metabolic changes. Even smaller, you go for immunological changes and then genetic changes. Biophotonics can probe concentrations that are close to immunologic changes. So you would be able to see very low concentrations around nanomolar, and there are a number of molecules that are detectable for using the optical technology. So if now let's go to the topics, the main topics of the biophotonics field. So I'll give you, we will discuss here about fluorescence and microscopy. We go to one of the most used concepts in biophotonics, that is diffuse optics. We discuss some of the medical applications of diffuse optics and go to the future trends in the field. So if we start with fluorescence and microscopy, you may remember that a conventional microscope is built like this. So there is a light coming from the bottom, and this light will pass through a condenser lens that will focus the image in a certain plane of your sample. So you have the image that comes from that area, and this image is magnified by an objective lens and then by an ocular lens, and then you get to see it. So microscopes can be coupled to fluorescence, and you can see certain phenomena such as cell division as pictured here in the GIF on the right side. So you can see there the nucleus that is the cell nucleus that is disappearing, the DNA that is going to each edge of the cell, and then the division in two daughter cells afterwards. So um, fluorescence in this case is really used to enhance your images, and you can see many details that you couldn't see in conventional optical microscopy before. So not only the details, but you could see even structures that are not appears like a blob, as you can see in the right side, but it is actually more more structures if you see in a fluorescence image. So because in fluorescence we don't have problems with background light, then there are several advantages with more sensitivity, better contrast, and it's possible to label multiple cellular structures at a time. So in this case, there are several ways that we can probe biomolecular processes inside the organisms. And to do this, we can use several types of molecules. So we can make the organisms to produce um, several types of proteins of different colors and track these proteins. For example, inside a cell, if we go to a bit bigger size, so we can check or we can label the cells of a mouse brain, one type of each color. We can even probe the organs of an organism or map the entire nervous system of a zebrafish. And all of this was possible because of a technology that is with the green fluorescent protein. And the discovery of this technology actually gave the discoverers, the researchers who discovered those, a Nobel Prize in 2008. So what they did is that they found a DNA that produced the GFP or green fluorescent protein in jellyfish, and by looking at this DNA, they were able to have this sequence and modify the DNA of an organism so that they would produce this GFP, and those which produced this GFP had the organs that produce the cells that produce these proteins as fluorescent. So this revolutionized the whole field of biophotonics, and other other discoveries are with precise localization or single molecule information. Um, now, if you it is possible to see even the pores of the nucleus of a cell. So these are single molecule information. If you see the figure in your left side with confocal microscopy, which basically means you'll be focusing a beam of light in a small point of your sample at each time, and then you scan the image. So with confocal microscopy, it's not possible to see these pores. But if you use super resolution microscopy, it's very well defined. So you can see even the structures, the outer structure with the basket or the ring of this pore. And this is something that is of a size of a wavelength. Usually, if you have the best instrument in the market, you will not be able to focus light in a very small volume, in a volume that is of the size of in a distance that is of the size of a wavelength. It's not possible to focus light because of the diffraction limit of light. But how to overcome that limit if it is not focusing? So with super resolution microscopy that these people have discovered. So one of the technologies is with super resolved fluorescence microscopy, and especially one of them is stimulated the emission depletion microscopy. So what happens in this example that I will give you is that we have some green light coming through a lens. So imagine that you have this light focused in the in the sample, and you have two beads that are separated by a wavelength they are a wavelength distant of each other. So when you use your laser beam to probe these beads, then you actually see the rise in intensity, and once you pass the beads, you have the intensity go downwards. So you actually see those two beads as one blob. But if you create a donut where in this donut fluorescence doesn't occur, fluorescence occurs only in the center of the donut where there is no light, no induction there. So when each bead passes through the center of this donut, then you see a rise in intensity, and you are able to distinguish the two beads, distinguish the 2 points that are a wavelength separated from each other. So this is the way that they did one of the techniques of super resolution microscopy. And this is already widely used worldwide. It changed completely the way we do things. And still in the microscopy world, it's not, we cannot only see small structures, but we can also manipulate them. So there are techniques with micro manipulations and with optical traps or optical tweezers. In this case, the beads are concentrated in the focus, and you can use this to manipulate your sample, for example, by self-guiding all red blood cells into a certain area of your sample, or even building micro machines that will control the flow inside the cells. So all these optical traps and optical micro manipulation technology gave Arthur Ashkin, one of the Nobel laureates of 2018, his Nobel Prize. So for the discovery of the optical tweezers and their applications in biological systems. Now, if we switch a bit from the microscopic world, and we go to a more macroscopic scale of tissues, then we have optical clearing methods. In this case, we are decreasing the light scattering that happens in tissue and making it more transparent. So with this technology, it's possible to image an entire organ, for example, an entire skull of a mouse or an entire brain, and you can see even cellular structures. You can see all the cells inside the brain and map all of it. So this is something that was really recent. You can get even images and videos of several biological processes, and I will show actually some videos here to you.
Being able to observe a neutrophil just crawling through the ear of the zebrafish, scooping up all of these food particles that we've injected, that's just mind-blowing. I actually got goosebumps in my hair. I still have goosebumps thinking about the moment when we actually first saw that. Being able to visualize that was phenomenal.
Cancer cells. So these are human cancer cells that have been implanted in the zebrafish, and you can see now the cells moving along capillaries, cells crossing the wall of the capillaries. But it's not just a blob of the cell like other people have seen in the past. This time we see the details of how the cell is sending pieces of the cell to explore their space.
We've mapped the entire eye of the zebrafish and then computationally exploded the cells such that you can look at each cell as a unit, and what you can actually observe is the changes in morphology in the context of the tissue. That's never been done before. You can look at how all of the intercellular compartments are dynamically changing, right? We can see cell mitosis, cell division, cells kind of pushing against, beating up their neighbors while they're kind of violently dividing, or at least that's the impression I get when I'm looking at the cell divisions.
Now I cannot sleep anymore. I'm up every night trying to figure out how to do the next thing.
I will show you just another one with the mouse brain. So this is a map of how you can see inside the mouse brain. You can see all the cells there. So yeah, you are just we just see brief just a brief overview of what is the potential of this technique. So as you see, this type of technology has a lot of applications, and it actually is used to not allow the researchers to understand the molecular origin of diseases, but also many other processes, many other biological processes. So if we switch to a more macro scale, then fluorescence is used for diagnostics. And in this case, we are using the tissue level. So fluorescence diagnostics can use contrast agents. In this case, the structures are very enhanced, so there's an increase in sensitivity, and you don't need very complicated image processing algorithms. But you have the side effects of the injecting dyes, and the doctor should perform the clinical procedure. There are other approaches that don't use the contrast agents. These are the label-free approaches, and in this case, there is a completely non-invasive technique that you can use anytime for how long you want. In this case, there is more time to process the captured signals, and in order to obtain relevant biochemical or biological information, but it can be used anytime. So let's discuss a bit of these two. So if we start with the ones using contrast agents, one of the most known one is using the ALA, or this is the abbreviation for aminolevulinic acid. And in this case, the fluorescence comes from another compound. So in this case, ALA is a natural compound that occurs in our organism. It is used to produce protoporphyrin IX, and then the heme that is used for the hemoglobin that is in our blood for picking all the oxygen and distributing the oxygen all through all the cells of our body. So we actually survive because of this. If we administer ALA externally, and then we can wait for some time that some protoporphyrin IX will build up in your system, and it will accumulate preferentially in the tumors. And protoporphyrin IX has actually a red fluorescence, a strong red fluorescence, so that when we illuminate the tumor with a blue laser, for example, it will shine in red. So for diagnostics, if we want to locate the tumor, we will probe for a specific peak at the wavelength of 635 nanometers. So this is where we have the red emission of fluorescence. And protoporphyrin IX is also something that makes the cells more sensitive to light. So if we illuminate the cells with a red laser, it is possible to kill the tumor cells. So we do both diagnostics and therapy at the same procedure. It's obviously possible to check again if there are any other cells that are remaining and other tumor cells that are remaining with the blue laser again. So this is one of the procedures that is performed. And if you see a spectrum, which means the intensity as a function of wavelength for the normal tissue, then you see only the fluorescence. Only the fluorescence of the normal compounds in tissue. But if we want to look for the emission of the tumors, then it will have a big peak, a large peak of red fluorescence, like this one with the malignant tissue. Now, if we compare the two approaches with contrast agents and without it, so you have the PP9 fluorescence by using ALA in your left side, and an approach without any contrast agent in your right side. So we are looking at a brain tumor, and we look first at the spectrum of normal tissue surrounding the tumor. It looks like this. If we go to the border of the tumor, it will have that huge red fluorescence emission. And then inside the tumor, it will be always with the strong fluorescence emission too. So if now if we switch to something that doesn't have the contrast agent, the normal tissue looks like this. The border of the tumor will have some protoporphyrin IX, but will not have the peak of its emission will not be as strong as if it has the induced protoporphyrin IX from ALA. And in this case, if you go to inside the tumor, there will also be some of the production of PP9, not as strong as well. So you can process that signal and get where are the areas that the tumor are. So we can probe the tumors with spectral techniques. In this case, with point monitoring, and we collect the intensity as a function of wavelength. In this case, we are more molecularly specific. As we can also do imaging. In this case, we have less spectral information, but it is also in a larger area. So we can always use the two applications for the more, we can always use the application that is more, the technique that is more useful for each application. And then here, I'd like to just make a brief break for questions. So do you have any questions so far? So yes, the first question is from David. It is, how can you kill cancer cells with red lights? So what happens is the protoporphyrin IX has an absorption of red lights, and this red lights will allow the protoporphyrin IX to go to another state where it can transfer its energy. It actually goes to a triggered state, and then it will transfer its energy to other molecules such as oxygen, or generate reactive oxygen species. When you have these species in the cell, they react with almost everything in there. So basically you are destroying the cells by oxidizing it from inside, oxidizing them from inside. And the way when you just illuminate the cells with red lights, and then yeah, you just kill them from inside by exciting the protoporphyrin IX. Yeah, is there any other questions? Okay. Yeah. So let's just proceed. So we will have more time for questions in the end, but let's just proceed for now. So as I said before, with fluorescence, we don't expect to have a lot of background light. So it may be affected by background light. And if we want to avoid those effects, or if we want to use other techniques that are not affected by background light or environmental light, then we need to understand a bit of diffuse optics. So this is something that is very widely known in biophotonics. Let's go over it. You may have seen, okay, you may remember your linear optics course where you have a medium with refractive index at a 2, surrounded by a medium with refractive index at a 1. And in this case, the incident light may have some specular reflection in the first surface. It may go through the medium with direct transmission, or it may have some total internal reflection there inside the medium. However, in diffuse optics, this doesn't really happen. There is not such thing like a direct transmission, because we are not transparent, right? So in diffuse optics, so we have a lot of the light is totally scattered inside the medium. This means the light bounces many times until it reaches the other side. And with diffuse optics, if you compare, for example, a laser beam passing through 2 cuvettes, one with a clear medium and another one with a turbid medium, then you see the light is diffused all over the medium. I show you a GIF here in the right side. So this is the top view of a glass of water and some of the light scatterers. Once you increase, and there's a laser pointer that is pointed to the other side of the glass. Once we increase the light scattering in the medium, you see that it's very unlikely that the light of the laser beam, of the laser pointer, will reach the other side. So actually the light becomes very diffused all over the medium. And this is what happens in diffuse optics. Diffusion can be found almost everywhere. You can see this in the clouds in the sky, or if you go to the beach, just remember the last time you have gone to the beach, you may have seen the diffuse optics happening there. And you may not have noticed for the first time, but maybe especially because the last time, if you were planning to go to the beach this year, I hope the COVID doesn't stop you. But just remember that the next time you go to the beach, you can see all the diffuse optics beauty there. So, but diffuse optics is also present all over the nature. So you can see diffuse optics everywhere. We are diffuse. So if you see your hand, for example, you'll not see your bones inside your hand. This means the light will be scattered in your fingers. So this when light propagates through the medium, it's not like X-rays. You'll not see, for example, your bones with a clear shadow on the other side. If you have a tumor, for example, in the medium, you'll not see a clear picture of the tumor there. The light may actually go around the tumor, and then you see a diffuse shadow in the other side. Just to show a quick demonstration of this, let's go over to Sanatana Konoglu, who is a postdoc at the Biophotonics group at Tyndall and IPIC too.
Good afternoon, everyone. So it was a very interesting talk, and you would have enjoyed and you would have seen various facets of how biophotonics works. Okay, I hope you are enjoying. Now, there is big challenge, because if you see there is MRI and there is CT scan, all this has been explored around 1970s and 80s. They have created a breakthrough. What's that? It's tomography. So if you have a human body, you can scan across the human body and you can find all organs and bones and all the tomographic structures of each of your cell, each of your organs by using a X-ray. But by the way, we all know that X-ray is carcinogenic in the sense they can mutate your genes and can create cancer. As we move forward, we are looking for alternative. We are always trying to build a world that is much better. And so I want to show you here how we are different and what are the challenges that we have as compared to typical tomography, because one of our targets is to make tomography. When you say a CT scan, what it does is it has a camera. Imagine your eyes as camera. So when you see this way, you see the knife that has this nice parallel structure and there is a rectangle structure. But when you move this around, it becomes a straight line. So that's what is tomography. What happens is that, as you can see in the slides as well, there is a cone structure, but when you see from one angle, it's a triangle. When you see from other angle, it's a triangle. When you superimpose both of them and rotate this phenomenon across, and when you just back project them, you get the 3D structure. This is what happens in tomography. So I will start my demonstration with how an actual tomography works by trying to collect tomography of this particular knife and on the wall, where you can see the shadow of this knife at different angles, 0 and 90 degree. And I will show you what happens when it becomes an actual human body, because actual human body, as Marcelo showed, nature is diffusing. You have something diffusion, that means you can't see through. And that's where I am going to end, and I am going to tell how we can overcome the challenge. So as you see, the perfect shape of the rectangle of the knife is seen nicely. Now what I'm going to do, it's actually a water bottle. So you can see the knife edge, and also you can see the water bottle, okay? And I'm going to do a kind of tomography by rotating the knife. Okay, so I'm just going to rotate the knife. As you see, that rectangle is going to become like a straight line. Now it's a straight line you can't even see, simply because you are looking at the other dimension of the knife. So again, I am going to rotate by 90°. Please look at the wall, not in the bottle. Okay, look at the wall. And it's a straight line. Again, when I rotate it back, it's again, you can see the clear features of the knife. Okay, so like this, if I take the picture of the wall at all the angles, and if I try to back project it, what's going to get is I'm going to reconstruct my knife. This is not the challenging thing. This is what all the X-rays does. The real challenge with diffuse optics is we are diffusing. So I am going to simulate now a human body by adding a diffusion coefficient. That is, I'm just going to add the milk. Even before I go there, now you can look at the bottle. I am sending in as well the laser light, okay? But you can't even see the laser light inside the bottle, can you? No. That's because water is transparent. The light just goes through the water. Now I am going to add, as you see, a little bit of milk. Just see what's going to happen to the shadow. Okay, and also you can see you see the milk is getting a bit of bigger. I suppose the camera got frozen. Sorry guys. Yeah, can you see again? Okay, probably. So as you see, it's getting more and more messier, and if I put laser light, it's scattering everywhere. Probably you can't see. That's because now I am adding milk to the body, but still you can see to some extent on the wall still the signature of knife, if you can see. Okay, still you can see the shade of knife. It's getting blurred. What happens if I add more milk to it? Now it's completely lost. This is exactly what happens in our human body. Here in milk, we have lots of milk particles, but in the actual human body, what we have is red blood cells, organelles, and lots of things which create these kind of microstructures. Okay, that's what makes the scattering happen and not able to see anything at all. We are not able to project anything at all. We can't see the bones at all. Now getting back, I want to just mention towards the end, what's the actual thing about all these things. This is really messier for us because we can't work in biophotonics like this. But if you see on the other hand, it's the great challenge. If you are able to sort the challenge, you already saw a picture of microscope innovation, which has brought many people Nobel Prize. And here it is, here is the innovation. Can you develop a technology that could see through the human body? Yes, we can. At Tyndall, we are working towards it. We have developing all the possible models and algorithms that understands when you shine a laser or a simple light into a human body, in which path it takes. You understand? And each photon, the direction that it takes is very well understood by us. And if you know what direction it takes, you can always compensate and reconstruct the human body. That means you can see through your body. That means we could very soon, probably maybe in a couple of years, will attain what has been attained by X-rays. If you are interested to be part of such an exciting journey, there are many reasons. There is one such reason that you could try to choose this exciting field and work through it. Thank you so much. If you have any questions, please feel free to drop a message. We can discuss more on it. Thank you. Thanks, Marcelo.
Thank you, Sana, for the amazing demo and the amazing explanation. After an amazing demo, so let's discuss a bit more about tissue optics and come back here. So as Sana said, this is one of the huge challenges in biophotonics. So you may see that in tissue optics, it's the figure you have is a bit different from what you saw in your linear optics course. So instead of having the instant light that's only specular reflected and being directly transmitted inside the biological tissue, the light will get totally scattered inside and may even come back in the other side. It may also go through the medium, be scattered, and then absorbed afterwards. It may be also absorbed, and then there is some fluorescence or phosphorescence or Raman scattering emission after that. So it's an emission with a different color of light than the one that you are using to illuminate the medium. And the light can also be scattered and be transmitted afterwards. So there is a whole range of processes that happens in biological tissues, and we can use all those to understand better what is happening there in terms of biochemistry and structure. So if we look at the tissue absorption, so here I will show you the absorption as a function of wavelength. So if we see the main absorbers in tissue, these are the blood in the visible range, and in the near infrared range, we have the water absorption. These are the main absorbers. There is a gap where there is almost, where there is a very little absorption. And this gap is called optical window. It's where we have a lot of the transmission of the lights. So if we are looking to the absorption, for example, you may remember the Beer-Lambert law, where you have a transmission from one intensity of light that is in one side, and some of this light is transmitted in the other side of a medium. So if you take, for example, a medium that has 1 centimeter minus 1 of absorption coefficient, then after a centimeter you have attenuation of 2.7, for example. If we compare the absorption from the blue, so at 400 nanometers, and the red absorption, you have actually 1,000 times more absorption than the red region. And if you compare intensities, you actually have the intensity of the blue, the transmitted blue light, is actually a decimal with 400 zeros less than the red transmission. So as you may think, the wavelength matters when you are looking at at light penetration in tissues. And if you look at this, how much scattering will attenuate the transmitted light, it is around here. The typical value is around 10 centimeters minus 1 for several wavelengths. So as we discussed here, the wavelength matters for the light penetration. And if you have an example of laser pointers, you may use them to see how much light is transmitted through your finger. The blue light of a laser pointer will not have any transmission, but a red light will you may be able to see the red light in the other side of your finger. And that's why we use the red light to probe the oxygenated and deoxygenated blood, for example, for pulse oximeters, to know the blood oxygenation through the absorption of the blood in your finger, and also monitor all the changes despite all the minimum concentration of blood that is present there, since blood has such a great absorption. So, but we don't target blood not only for diagnosis, but we target blood for like therapy too. So if you remember from the photobiomodulation, we can target the blood, we can use the blood absorption to target the vessels and cause vasodilation, which will lead then to anti-inflammatory responses and all the benefic effects of those. So if we go to the medical applications of diffuse optics, then one of the main ones in the biophotonics field would be with diagnosis. And usually diagnosis and all the applications will be based on, most of them will be based on the tissue constituents of, and these would be not only blood and water. We can have a biochemical profile of lipid, collagen, tyrosine, or thyroglobulin. There a number of molecules that we can target, and by knowing their absorption, and we can, if we measure their absorption, the absorption of the tissue, we can fit the spectrum and then quantify how much is the concentration of each biomolecule inside the tissue. So we have actually a biochemical profile of the tissue without even changing it. We just illuminate the tissue, and we know the profile of many biomolecules that are present there. So this is one of the beauties of the biophotonics. So this is used for many applications, including disease detection in many parts of the body, as Sana was commenting to you. So this is used in many parts, either breast, abdomen, thyroid, bone, but all for head and neck cancer, gastrointestinal tract. It is applied really to all the parts of the body for many applications. Disease detection is a very good field to be in, and it is also used for surgical guidance. You have all the biochemical profiles during surgery, so the surgeon would be able to take the best actions, the best decision making by knowing these profiles. And it is also used in functional endoscopy and colonoscopy. So just to give you an example in the diagnosis side, so if we probe breast cancer and we look at the map of the tissue constituents, you might see the map here in white and black, where you see in the center that there is a tumor. And if you see the profiles, the biochemical profiles of the oxyhemoglobin and so on, you see that the tumor has a different constitution compared to the normal tissue surrounding it. So it is possible to locate the tumor and also provide the biochemical profile at the same time, just by illuminating the breast. So as I said, there is an enormous list of applications, and this will be discussed further in the next episodes. So in the next webinar, we'll have Professor Anderson Engels, who is the head of our group, and he will talk more about disease detection and surgical guidance topics in the next week. Just don't lose this webinar. It will be extremely interesting. So not only for diagnosis applications, knowing the absorption of the tissue constituents also allows us to see how we can propose treatments for diseases. And one of them is hyperbilirubinemia. So this happens in 60% of the babies born. It's very common. It is caused by an accumulation of a molecule called bilirubin. And it is a yellow molecule. So it is generated by the breakdown of red blood cells, and it is toxic for the babies. It can, if it is not able to be removed, then it accumulates in all the organs, including the skin. So the baby looks very yellow. If you look at the absorption of bilirubin, you see it absorbs a lot in blue. And this is why there is a treatment with phototherapy that uses blue lights to break bilirubin, and then lets the babies to remove this from their body. So let's see a video on that.
Jaundice is a common condition in babies.
We can see the screen.
It presents as a yellow discoloration of the skin. This yellow discoloration is a buildup of bilirubin. Some babies with high levels of bilirubin will require treatment. The most effective treatment for babies with jaundice is phototherapy. Phototherapy involves a shining light both below and above the baby, and this light changes the bilirubin from being dissolved in fat in the body to being dissolved in water, and therefore excreted in the urine. Babies admitted to the intensive care unit requiring treatment for jaundice will require blood tests to ensure the bilirubin level falls, and as these levels fall, the lights are then removed from the baby. When the level is below the line that is requiring treatment, the baby will be able to be discharged home. Phototherapy is an effective treatment for jaundice.
This was one of the videos of how we can use biophotonics to help the babies all over the world. It's we use, by knowing the absorption of the naturally occurring compounds, we can propose treatments. We can target also the absorption of not compounds that are not naturally occurring in the body. We can genetically modify organisms, and if we look at their absorption, we might propose, for example, looking at the functions of the brain functions. So this is what is exploited with optogenetics. In this case, it's a bit different from the stimulation, from the neuron stimulation we proposed, that was proposed in the past. So with electrical and light stimulation, usually you have an area that stimulates all the neurons. But with optogenetics, you modify the organism genetically so that some neurons will produce a certain molecule that will have a specific absorption, and you can either stimulate or inhibit a particular type of neuron. By doing this, you can also stimulate certain functions that we could never stimulate before. So we can probe selective neurons at a time and see the response of the organism. So again, this is one of the videos of a mouse that where the sensation of thirst was induced through shining light in their brain. So in this case, the rat or the mouse is just going around the cage. When the light is on, then it feels very thirsty. So it goes to the tap of water and then it starts to lick the water. When the light is turned off, then it starts to go around the cage again. And there will be, at some point, the light will be activated again and it will feel thirsty again and go to the tap. So this is one way of how selecting several neurons allows you to stimulate certain brain functions in animals. It may be used in the future for humans, and then in this case, there might be a way that we insert, for example, viruses in that will modify the DNA of a person, and then you can stimulate certain functions of the brain, or you can just study many of the brain functions by stimulating the selective stimulating the neurons. So now let's talk about the future trends in the field. So you might have, if you have watched the Star Trek before, one of the aims of biophotonics would be obviously having the tricorder, where if you scan your body, you would know all the diseases you have in one go. So we are going to that future. We are moving towards that. One way that we could do that would be combining optics with ultrasound, and this would be would combine the contrast that we have with light-based techniques. So the contrast that is provided by looking at the biomolecule profile of the tissues, the differences in biomolecular composition, and the resolution, the deep resolution in tissues that ultrasound brings to the field. So since light cannot penetrate very deep into tissue, and ultrasound can, so it's possible to combine light and ultrasound to achieve both contrast and resolution. And one example of this is acoustic-optical tomography. So in this case, the laser, a laser is used to illuminate the same point where the ultrasound is focused. And at the ultrasound focus, there may happen a frequency shift. So the optical detector would detect mostly the radiation from the laser, but there would be some radiation with a frequency shift of the ultrasound. And if we tag these, it is possible to image up to 18 centimeters deep, which means it is possible to have a biochemical profile of the whole body. So we are moving towards Star Trek, or tricorder at least. And another way that optics is advancing is looking. There are very small devices where we can look at the biochemical profiles of even inside the blood vessels. So this is the biochemical profile we have of coronary arteries. And inside the arteries, you would be able to see all where collagen is located, where lipids are, or elastin. So if you have any part where you would have a blockage due to either out of cholesterol or other diseases, atherosclerosis and so on, so you would be able to see them with this kind of imaging, and you see the biochemical profile in a very small detail of blood vessels. So not only this, it is possible to have augmented reality. If the surgeon wears a glass, for example, it is possible to see, to wear a glass, and while looking at the image, the surgeon can select an area to probe and look at the spectral features or the biochemical profile of the interest, the region of interest in the surgical field. So the surgeon can just choose an area and look at the biochemical profile of that area. And this is already ongoing. So this technology is upcoming. So you might expect this in the next years. And there is also a push for miniaturized instrumentation. So a lot of what we do in Tyndall and IPIC, as you may have heard of. So there is a push for wearable devices, not only smartwatches, but you may have something in your socks or in your clothes, or even in headsets. So you'd have very tiny microbiochemical sensors, and it is also possible to integrate all these technology in the medical equipment, even in the tiny tips of an endoscopic probe. So also there's a lot of microfluidic devices, so you can do chips where you don't need to go to a laboratory to a blood test, for example, and you could probe a lot of diseases by carrying this small chip with you. You can bring this everywhere, even when where people don't have so much money to buy these things. So we could increase the number of tests, the health tests that we do worldwide. And this is one of the best potentials of biophotonics. Miniaturized technology is also present is also being tried to be integrated in the smartphones. So with the spectrometers and microscopes. So if you remember the spectra that we were showing you, that spectra can be integrated into smartphones, or microscopes can be used with your smartphone to see cells and diagnose diseases on site, and just by even counting cells with your smartphone. So this is another upcoming technology, and we can use all this range of technologies, miniaturized technologies, for telemedicine. So everyone would have a device. They would be able to see, to monitor their own health, their own personal health, and then communicate to their doctors for a response and advice. So this would, for example, being prevented a lot of issues there during the COVID outbreak. And this is one of the main contributions of biophotonics. This is happening a lot and starting to have more and more importance. So all the advances towards telemedicine are now increasing and booming. So I hope now I have convinced you that biophotonics is widely present in our daily lives, and it has applications in both micro and macro scales. It is widely used for disease diagnosis and treatment, and it has potential to revolutionize healthcare. So if you are wondering about all the references that were used in this presentation, they are all here, and we will make this presentation available to you, and also we'll have the recording available online. So if you want to contact me, also these are my contact details.